Bonjour tout le monde. Hello everybody. This is the second day of our meeting, and I am Pascal Gerbeau, and I am pleased to just explain in 2 minutes how things will go. So we have 2 sessions with several speakers. There will be a break at lunchtime, a second session, and a final session. I would like to insist on the fact that for many reasons we have to run this meeting virtually, but there will be the possibility to ask questions through the internet. So the questions will be passed to the speakers by the chairperson, and we will have the possibility to have the speakers to answer. There is a special session at the end of the day, which is human genetics. The talk will be given by Jean-Louis Mandel, and it will be followed by a roundtable with speakers who live in Paris, so several speakers, also with the organizers, and very especially with students who have accepted to really ask their question to the speakers. So this roundtable will be in French, and we are very happy that the students will ask the question. So thank you very much for being here, and I will leave the stage to Jean Wessenbach, one of the co-organizers, to present the first speaker.
Thank you, Pascal. Good morning, everybody. We will start now with session 2, molecular traces of ancient adaptations and selection in ancient and modern human DNA. And our first speaker will be Luis Quintana Murci, who is a professor at the Institut Pasteur and since some time also at the Collège de France. As all the speakers, Luis has a long-standing commitment with human evolutionary genetics, first studying migration patterns, where he made very interesting contributions about the roots out of Africa. And at present his major focus is on the interface between immunology, evolutionary genetics, and he made some major findings in this area in the recent past. He was also recently elected as a member of the Académie des Sciences, and he will talk now on the demographic and adaptive history of Homo sapiens with a focus on Africa and the Pacific. So please, Luis.
So thank you very much to the organizers to invite me today to give this talk on how humans during their dispersals have adapted to different pressures. And today I'm going to focus on 2 main regions of the world, that is Central Africa, and more recently we have become interested in the Pacific. So as you have heard yesterday and today, we all know that humans originated in Africa 300,000 years ago, but the most important thing I want to highlight today is the last 60,000 years of human history that was based on major dispersals around the world. But the most important thing is that these dispersals were accompanied to adaptation to different climates, to different nutritional resources, and also, in the context of what interests us the most, to different pathogens. So this is the basis of biological adaptation, host biological adaptation to pathogen pressures. So let's start with our studies in Central Africa. So basically, why Central Africa? Because as you heard yesterday from Matthias Jacobson, one of the major migrations in human history started in West Central Africa, in the region that is today in between Cameroon and Nigeria, associated with the Bantu expansions, the expansion of agriculture, and iron usage. So Bantu speakers dispersed all over sub-Saharan Africa over the last 3,000–4,000 years ago. And today, there's a few pockets of surviving hunter-gatherers, such as Central African rainforest hunter-gatherers, usually known as pygmies because of their shorter stature. East African pastoralists, and also Khoisan populations in South Africa that Matthias also mentioned yesterday. So the questions we have been addressing over the last years have been to try, through genetics, to reconstruct the demographic history of both farmers and hunter-gatherers, to reconstruct the early steps of the Bantu migrations, to understand how these migrations of Bantu, that they encounter completely different environments, rainforest, desert, how they rapidly adapted to the new environments they found, and also finally try to understand how humans have adapted to the extreme conditions of the rainforest, of heat, humidity, and pathogens. So over the last 10 years or so, we have been studying these two populations in collaboration with the Musée de l'Homme, how they have interacted, farmers and hunter-gatherers. And to cut a long story short, here you can see in this tree the history of these two populations. You have seen that hunter-gatherers that are highlighted like RAH, rainforest hunter-gatherers, over the last 100,000 years of their history, their population sizes have been as important as the ones of farmers. We have also seen that pygmies started to separate from farmers at least 100,000 years ago, being this the second most divergent population split in human history, the first being the one with Khoisan populations, as was mentioned yesterday. And lastly, if you focus on the last part of the tree, in the bottom, the last 20,000 years of this population has been characterized by strong bottlenecks in hunter-gatherers and massive expansions in farmers. But what about farmers? One of the questions was to reconstruct how Bantu migrations occur in Africa. That's why we studied a few years ago around 2,000 individuals from 57 different populations at the genome-wide level. And we particularly focus on the homeland of Bantu speakers, that is, this region in between Cameroon and present-day Nigeria. And the reason is that there has been two hypotheses to explain the expansions of Bantu speakers. One is called the early split hypothesis that postulates that right from their homeland, they split in two different migrations, one to East Africa and another one to South Africa. Whereas the late split hypothesis postulates that they first traverse the rainforest, the equatorial rainforest, to eventually split towards the east and towards the south. So by comparing the genetic diversity of East Africans with the homeland of Bantu speakers and of South African Bantus to the homeland of Bantu speakers, what we have shown is that all Bantu speaker population resemble more to the genetic diversity of people that are situated in the south of the rainforest, supporting the late split model by which Bantu speakers first traversed the rainforest to eventually split into migrations. But during these migrations, Bantus encounter local populations of hunter-gatherers, like pygmies in Central Africa, pastoralists in the east, and Khoisan in the south. Here you can see an admixture plot. And how do you read that? Here there's 2,000 individuals. Each little line is one individual. You can see that there is a big red cluster. These are all Bantu speakers. But you can see that there is a part of Bantu speakers that they show each individual a green genetic component. And this green genetic component, if you see in the right part of the panel, you see that comes from pygmies. Basically what this shows is that Western Bantus admixed with pygmies, Eastern Bantus admixed with the local populations they found in East Africa, that is pastoralist, and Southern Bantus admixed with Khoisan populations here, the genetic component being in yellow. But most importantly, what Etienne Patin in particular discovered is the concept of adaptive admixture, What does it mean? It means that when Bantus encounter the pygmies, while admixing with the pygmies, they acquire beneficial mutations that allow Bantus to better survive, for example, in the rainforest. For example, Western farmers, through admixture with pygmies, they acquire beneficial mutations at the HLA region and at a gene called CD36 that is involved in resistance to malaria. Eastern Bantus, while they admixed with East African pastoralists, they also acquire the beneficial mutation that today allow most of the populations to digest milk in adulthood at the lactase gene. But this adaptive admixture can be both ways. So this is why we revisit a kind of iconic case of balancing selection in humans, that is the sickle cell allele. As you all know, in heterozygosity, the sickle cell allele is associated with protection against falciparum malaria, despite at the homozygote state leads to severe sickle cell anemia. So the rationale here was, because if malaria was not there, therefore there won't be any selective advantage, this allele will be eliminated by negative selection in 12 generations, because at the homozygote state is associated with severe anemia. By dating the time of selection of the allele is a way of approximate the time at which malaria started to be a strong burden for the population. So to understand this, we sequenced the hemoglobin gene in around 500 individuals, farmers and hunter-gatherers all over the Central African rainforest belt. And also again, to cut a long story short, what we found is that the sickle cell allele originated in the ancestors of present-day farmers, that at the time obviously were hunter-gatherers, 25,000 years ago. So pushing back of at least 5,000 years the origins of malaria in Africa. And most importantly, what we show is that farmers transmitted the sickle cell allele to hunter-gatherers only 6,000 years ago through adaptive admixture. What does it mean? This is called the poison antidote hypothesis, meaning the Bantus, they encountered the pygmies during their migrations, they started to deforest the rainforest, they brought malaria to the pygmies, so the poison, but they also brought the allele that allowed the pygmies to resist malaria, the antidote. To finish this first part also, we have been quite interested in understanding human adaptation to the extreme conditions of the rainforest. As you can see here, in all regions of the world, there is a classical phenotype that is associated with the rainforest, that is shorter stature. So we have been quite focused with pygmies, what's called the pygmy phenotype, meaning shorter stature, in Central Africa. And until a few years ago, many claims and conclusions about pygmies were based on a single population of pygmies as representing the whole pygmy genetic diversity. That's why we started a project also in collaboration with many people, including here in Paris with Paul Verdoux from the Musée de l'Homme, studying 7 different populations of pygmies from Cameroon, Gabon, and Uganda, together with the neighboring farming populations. Again, here you have an admixture plot, and in the left you can see all the populations of pygmies, and as you can see, there's more difference between 2 pygmy populations than between pygmies and farmers, showing that they show high genetic diversity among them. But the question here was, let's try to identify genes that have been under a strong positive selection in pygmy populations, because this will tell us about genes and functions that have participated in adaptation to the rainforest. So to declare that a function or a gene has been under positive selection related to the pygmy phenotype, what we did to avoid false positive is that the signal of selection has to be present in at least 3 different pygmy populations, and obviously absent in farmer populations. So we found around 30 different signals of genes that have been under positive selection in pygmies, but I want to focus today on the single signal that was shared among all pygmy populations, including the populations that are highly admixed. Is this gene that is called TRPS1 that shows a strong signal of positive selection and is associated with skeletal development, morphological features, and immunity. So kind of informing us about functions that might have participated in adaptation to the extreme environment of the rainforest. But not all these strong signals of selection. As you know, there's an increasing interest in what's called polygenic adaptation. What does it mean? In contrast with positive selection, that is a single mutation that provides a strong selective advantage, polygenic adaptation is many different mutations in many different genes, each mutation conferring a weak selective advantage, but additively conferring a strong selective advantage. So we searched for signals of polygenic adaptation in the rainforest, and we found there is signals of polygenic adaptation related to height, a phenotype that we do know that characterize populations of the rainforest, but also of other reproduction-associated traits, such as menarche and age of the first and last birth. But then we said, well, we know there's many genes that show pleiotropy, meaning a single gene might have different functions. So we say, if there is just one function that is the real function that is selected, and the other functions are just hitchhiked with. So to assess this, to investigate this, we remove all genes that have been associated with height. And we did the analysis again, and we lose completely all the significance of the other windows, suggesting that is height the real adaptive phenotype. And to ensure of that, then we test for height, and we remove one by one all the other reproduction phenotypes. And height kept being significant in all examples. So showing that height is highly polygenic, and due to pleiotropy of the genes that are involved in height, we can have spurious associations. So to finish, let me share with you a few minutes of our most recent results on Pacific Islanders. And why Pacific Islanders? Because the Pacific is a land of strong contrast. The Pacific was people for the first time just right after the out of Africa. 40,000 years ago, people reached near Oceania, meaning Papua New Guinea, the Bismarck, and the Solomon Islands. Then nothing happened for 35,000 years, and then just 5,000 years, populations from Taiwan, associated with the Austronesian expansion, come down through the Philippines, pass over Papua New Guinea, the Solomon Islands, that they were already people, and colonize for the first time the rest of Oceania, Vanuatu, Samoa, Tonga, and Polynesia. Don't forget that 1,000 years ago, there were no humans in Polynesia, for example. So to understand, and another thing that is very interesting of these populations is that they present the highest levels of archaic ancestry in the world. If we put together Neanderthals and the sister of Neanderthal, Denisovan, they are the ones that present the highest levels of this archaic material. So that's why we study 300 individuals, that we sequence the genome of 20 different populations, as you can see there in the right, of all regions that are supposed to be at the basis of the peopling history of Oceania. And we found 3 things. First, that during the settlement of Near Oceania was a strong bottleneck of the population. And the different populations of Near Oceania, Papua New Guineans, Bismarck Islanders, and Solomon Islanders, diverge already very deep in time, from 20 to 40,000 years ago. And second, that contrary to what's called the out of Taiwan model, that postulates that the Austronesian expansions started in Taiwan and very rapidly over the last 5,000 years peopled or colonized Near Oceania, what we found is that the Austronesian expansion was not followed by immediate admixture with Near Oceanians, because the admixture was just 3,000 years ago. Second, and one of the most interesting things, in terms of archaic introgression, we have found that while Neanderthal ancestry is very homogeneous across all populations, from 2.2 to 2.9%, Denisovan ancestry is very heterogeneously distributed, from 0% in Taiwan, for example, and in the Philippines, to 3.2% in Papua New Guinea and Vanuatu, for example. And most interestingly, what we have found is that while Neanderthals have contributed beneficial alleles to modern humans related to different phenotypes, neural development, metabolism, and pigmentation phenotypes, Denisovan has contributed beneficial alleles that are almost exclusively restricted to the regulation of innate and adaptive immunity. For example, interferon-regulated factor 4, that is, regulates toll-like receptor signaling and interferon responses to viral infections, suggesting that when humans reach the Pacific, by admixing with the Denisovans, they acquire beneficial mutations that they allowed modern humans to resist the local pathogens. And finally, what we have shown is that while Neanderthals is more clear in this summary tree, while Neanderthals here in red, there has been a single admixture event with the ancestors of Eurasians around 60,000 years ago, we have identified at least 4 different admixture events with Denisovans. For example, the last admixture events, we've dated them at between 20,000 and 25,000 years ago, suggesting that Denisovans may have survived until around 20,000 years ago. And finally, we have also identified adaptation signals to insular environments related, for example, to the regulation of anticonvulsive effects of endogenous pregnenolone during pregnancy, and interestingly also polygenic adaptation in the Pacific related or favoring lower HDL cholesterol levels. So with that, I would like to thank you, the people in my lab, the local communities, and our different collaborators for the African and Pacific studies and the funding agencies, and you all for your attention. Thank you.
Okay, thank you, Luis, for this crystal clear presentation. I think we all appreciate it very much. I have a few questions. The first one from Ludovic Orlando, who is asking if the timing of local admixtures is this timing similar, and does adaptive integration kick in right after admixture, or how long does it take before the introgressed alleles become adaptive?
Okay, that's a very good question. Well, actually, the admixture probably with Denisovans did not occur in remote Oceania, occur probably either in Southeast Asia, or probably at the time, don't forget that the sea levels were much lower. So basically you could walk from Papua New Guinea to Australia, was a single continent until 10,000 years ago. So what we have shown is that there is the 2 most important admixture events. Is one between Denisovan and East Asians around 20,000 years ago from a population that was very similar to the Denisovan Altai genome, the only genome we know so far. And the other introgression event is 25,000 years ago, probably in Papua New Guinea or nearby, but with a population of Denisovans that diverged from the Denisovan genome, so was a Denisova-like population because they diverged 400,000 years ago. When it started to be adaptive, this is a difficult question to answer. The fact is that they present very high levels of Denisovan ancestry, and as you know, in most of the genome, archaic admixture has been selective against.
Thank you. Another question is, what is the adaptive value of hate in tropical forests? How selection is realized on hate?
Basically, shorter stature is a way to thermoregulate your internal temperature related to sweat, to sweeten and to loosen energy due to the strong heat and humidity of the rainforest. It's a matter of surface for thermoregulation.
A complementary question about the last admixtures with Denisova. Was this after the expansion of in Oceania, or was it before? Do you have some insight on the geographic range of Denisovians in general?
Yeah, when admixture occurs, we have approximated through genetics in between 20 and 60,000 years ago. The geographic range of Denisovans, we don't know, but certainly in Tibet, in Siberia, because they have been found fossil remains there, and probably in Southeast Asia. Then we have no idea if the admixture event took place in Southeast Asia just before the peopling of Near Oceania, or if there were Denisovan populations in Near Oceania, because there are just no fossil remains so far.
Okay. Well, a very general question. Why human evolution started in Africa?
Origin of life.
Why? I don't know.
It's not a scientific question. To what extent does the variation in Denisovan ancestry reflect differences in the amount of Papuan ancestry in the
that's
ocean,
a very good question.
in the ocean population?
That's a very good question. Well, the interesting thing is that today we know very little about how diverse were the Denisovan, because there's basically just one genome of Denisova. However, by putting together all these 2-3% of Denisovan segments in modern humans, we can almost resuscitate who were the Denisovans. So we have seen, by putting together all these pieces of segment Denisovan in our genomes, that they were highly diverse. And I started to talk about another thing. Which was the question?
The question was the extent of the variation in Denisovan ancestry.
Yes, yeah, sure. Yeah.
And how does this reflect differences?
They are very diverse, because they are as diverse that when we compare these segments of Denisovan ancestry, we found almost different populations of Denisovans, because if we compare some Denisovan ancestry with other Denisovan ancestry, the divergent times is 400,000 years ago, which we call it Denisovans, but were maybe other populations of archaic humans. We have no idea, because they're just one genome and 2 fossil remains of Denisovan.
And how many Neanderthalian genomes you have?
I think there's complete and high coverage. There must be, Jean-Jacques knows much better than me, but I think 8, something like that, from 6 to 8, yeah.
Okay, I think we will proceed now, and
Thank you.
Thank you, Louis. And our second speaker is Johannes Causer. So he is at the Max Planck Gesellschaft. He's director of the Department of Archaeogenetics at the University of Jena for the science of human history. And he has made a number of contributions in Leipzig first, and some major contributions in the field, including Denisova, but also one of his main contributions was on the history of the plague bacteria, Yersinia pestis, and he will present us this fascinating story. His talk is entitled "The Genetic History of the Plague: What We Learn from Ancient Pandemics." I don't know if it's useful for the present day pandemics, but maybe.
Thank you very much for the introduction. I'm very glad to talk after Louis today because he has been focusing in the host-pathogen interaction on the host side and
adaptation to pathogens, and I want to focus today on the pathogen side and see what we have learned about the evolution of pathogens by studying ancient pathogens from the past, and with a particular focus today on the genetic history of plague and what we learned from those. How did, for example, tuberculosis look like 10,000 years ago, or how did plague look like thousands of years in the past? What is the mutation rate? So how fast do bacteria or viruses change in their genetic code over time? And also, what was the adaptation? How did, for example, a pathogen that was present in an animal population become adapted to the human as potentially a new host? And the main reason why we knew so little about the evolution of pathogens was that we didn't have fossils of pathogens from the past. Unlike for humans, where you might have Neanderthals or Homo erectus to look at, or for example, elephants, where you might look at the mammoth, we didn't really have pathogens from the past because often they don't preserve, you don't really see them, you can't really study them. And this is why we started this new field that we now call ancient pathogen genomics, where we decided to go straight back into the past and actually extract from ancient skeletons the pathogens that might have killed those people in the past, and then look directly into the evolution by studying their genomes and how they changed through time. And how we do that is basically taking ancient skeletons. Often we take teeth, because inside a tooth you might have dried blood. So a pathogen was blood-borne. You could then potentially reconstruct, drill in little holes, we release the DNA, and you heard much about ancient DNA already yesterday, and probably more even today. And then we use high throughput sequencing, next generation sequencing, to then reconstruct the genomes of those pathogens from the past. And that then really provides some sort of molecular fossil record. We can then reconstruct them, we can study them through time, look at mutation rates, look at host pathogen interaction at the pathogen side, but potentially also relate that to the human diversity, as Louis was just presenting this morning. And of course, we provide very important information for historians, for archaeologists, about the causative agents. So what type of pathogen caused an epidemic or a pandemic in the past? And I was doing most of that research together with 3 researchers, so Kirsty, Alexander, who are now group leaders at the Max Planck Institute here in Leipzig. So I'm not in Jena anymore. We just moved our department to the Max Planck Institute for Evolutionary Anthropology in Leipzig, but also Verena, who's now a professor at the University of Zurich. So much of our research was on Yersinia pestis, and I will talk more about that later today, but we also looked at the evolution of Mycobacterium leprae, so the causative agent of leprosy, even tracing it back now to ancient Egypt by reconstructing genomes from Egyptian mummies. We looked at the emergence of syphilis, so treponemal diseases, not just syphilis, but also yaws, and kind of relating that to also the transatlantic trade in the early 16th century. we looked at tuberculosis and found out, surprisingly, that before Columbus, tuberculosis was already present in the New World, but it wasn't the tuberculosis you find today in the New World, but actually a form of tuberculosis that was a zoonotic transmission from seals or sea lions to humans, which was a big surprise. We looked at Salmonella enterica, the causative agent of typhoid fever, which was one of the diseases introduced in the 16th century by the Spanish into the New World, causing probably large outbreaks, probably related to the so-called Cuculitzli pandemic. And we also were able, together with collaboration partners in Bolzano, to study the stomach content of the Iceman that was frozen for 5,000 years. We could actually reconstruct Helicobacter pylori genomes to know something about this pathogen that is present in about 30% of the world population today and how it changed over the last 5,000 years. But as I said, the main focus today should be on Yersinia pestis, where we've also learned the most of its evolution over the last few years, and also where we could trace this type of bacterial disease, not quite 50,000 years into the past, but at least more than 5,000 years. So when we talk about plague, we have to keep in mind it's actually not really a human adapted disease. It's actually a rodent disease that is found in wild rodent population like marmots or squirrels or other type of rodents in more than 50 different rodent species in the world. And it's transmitted not usually directly between those rodents, but there's actually a vector that transmits the disease, which is a flea. And the pathogen itself has adapted over the last 5,000 years, as you will see later, to transmit the disease by a very kind of nifty mechanism, if you want. So if for example, a flea then bites an infected individual, it takes up the bacteria into its stomach, and in the foregut of the flea, the bacteria are building a kind of biofilm, which causes a blockage so that the flea cannot swallow blood anymore. So the flea actually is starving because it cannot swallow blood, and it keeps on biting many, many times. So some of the researchers studying that type of fleas that have a blocked midgut, they actually bite up to 200 times a day. And every time they suck up the blood, it comes into contact with the biofilm, and then they release it again, infecting potentially a new individual, the same individual multiple times. So eventually those rodents will die of the disease of plague, causing a form of sepsis. And then the fleas are just looking for the next host, and that next host might be a mammal that is, for example, is living associated with humans, such as dogs, cats, but also especially rodents like rats or mice. It comes then in contact with human and human habitations, and then potentially also by flea transmission can actually infect humans. So the fleas then move in the lymph system, most often to the next lymph node that becomes big, becomes swollen, which is then called the bubo. This is also where the name bubonic plague is coming from. And then from the lymph system, the bacteria move into the entire body, infecting multiple organs, causing organ failure. And about 60% of all people, if they're not treated, it actually then causes death within about 7 to about 14 days. So quite rapid and very, very high mortality if untreated. And this is not the only form of plague. There's another form. So if people have, for example, then an infected lung, and they actually cough out droplets with bacteria, somebody else might inhale them, and again, it's directly infected into the lung. That's called pneumonic plague. So it's the same bacterium, but it's just a different type of transmission. And it first infects the lung, and that's actually a very severe form of plague with a mortality that is even higher. It is actually above 90%, and even treatment with antibiotics is very difficult because often the first symptoms occur after 24 hours, and death often occurs within the first 48 to 72 hours. It's still found in a lot of places today, in rodent populations mostly, but then often there's also then kind of due to zoonotic transmission found in human populations. We have about 2,500 cases in humans per year, which is compared to the past where it was really millions of people that got infected. But you can see that the mortality today is actually very low because we have very powerful antibiotic that can also, of course, be used for treatment. But if not treated, then the mortality, as I just mentioned, is actually quite high. Historically, there have been 3 pandemics that, based on the description from those early scholars that were writing about those outbreaks, would be attributed to Yersinia pestis. So one was the first pandemic, the so-called first pandemic in the 6th century, which is also called the Justinianic plague. It lasted about 250 years, and then in the 7th to 8th century, it just disappeared from Europe again. But it came back in the 14th century with the Black Death, the pandemic that's probably most known and most famous, because from what we know today, it's probably the largest pandemic that we know in human history. Again, it lasts for several hundred years, and then in the 18th century, it disappears from Europe, but it actually has a comeback, not in Europe, but in East Asia. So in the 19th century, in the mid-19th century, it emerges in the Yunnan province, and then from Hong Kong, it actually spreads all over the world due to shipwrecks and these steamships that are then connecting, of course, in this globalized world and all continents, and then it gets transported to Africa, South America, North America, and that again lasts for about 100 years or so, killing again millions of people. And then fortunately, because of antibiotics, and since the 1950s, it has basically disappeared mostly in human population, but still found in animal populations. And the third pandemic is also the time when Alexander Yersin was actually sent by Pasteur in the 19th century to investigate this disease in Hong Kong, actually found out that it's this particular bacterium, which was then also named after him. And he already found out then at the time that the flea is actually the vector that transmits the disease. I want to first focus on the second pandemic. As I mentioned, probably the largest pandemic we know in human history, with kind of the largest number of casualties. More than 50 million people died within a very short time. It only lasted for about 5 years, and it killed approximately half of all the Europeans, because it has this really high mortality. So probably almost everybody get infected, because as I said, the mortality is about 50%. So probably all of Europeans at the time got infected with this pathogen. So basically, they didn't have any safety precautions, so no face mask and isolation like we practice today. But it was a really, really severe pandemic, which has been historically reconstructed to have originated somewhere in the East. It has been suggested China, but the historical data really seems to show that it came somewhere from the Black Sea, so in Kaffa, which was a Genoese colony at the time. We have the earliest historical records describing that during a siege where the so-called Mongols, which was probably the Golden Horde Empire, was in a siege on this city of the Genoese people. They actually were catapulting dead people into the city, and it said that from those dead corpses, the disease was spreading. The Genoese had to retreat. They retreated on ships and took on those ships the disease into Europe, and then it spread all over the continent within this 5-year period. Yersinia pestis, as I mentioned, had been suggested, but about 10 years ago, when we started this research, there was also much debate about whether it was a virus, it was an unknown disease, it is something completely else, because the symptoms that were described for the medieval plague didn't really fit the symptoms that we see in the 19th and the 20th century during the third pandemic. So we then kind of collaborated with a number of people to try to find evidence, molecular evidence of Yersinia pestis from the past, but not just seeing whether it's present in the 14th century, but also seeing if we can actually reconstruct the entire genome. And this was quite challenging, because if you extract DNA from an ancient bone, the majority of that DNA is actually not from that person, and is of course also not the pathogen that might have killed that person, but it's actually environmental DNA. It's bacteria, plant DNA that has been deposited in the bone over the last few hundred or even few thousand years. So we had to develop methods to fish out the DNA that we're interested in, where we use hybridization capture. So we have little microarrays, so glass slides that have DNA on the surface, in this case, modern Yersinia pestis DNA. And then the DNA will bind the DNA that is complementary to it, which is those ancient tiny pieces of DNA. And you can then wash away all the DNA that has not bound, and by that enrich for the DNA that you're interested in. And that actually worked quite well. So if you look at the amount of Yersinia pestis DNA we had before enrichment, it was very small, and after enrichment more than 60%. So that allowed us to get the whole genome of Yersinia pestis reconstructed, in this case from individuals from London that were about 667 years old, so from the time of the Black Death, on a cemetery that was exclusively used during the Black Death. And one of the first questions now that we had was to see how this London strain from the Black Death is related to modern day Yersinia pestis. As I mentioned, it's found in rodents all over the world today. You can see that the majority of those strains are falling in those 4 different branches here, and that are called branch 1, 2, 3, 4, and then the other ones are called branch 0. And they diverged sometime in the past from Yersinia tuberculosis, which is the closest living relative. It's a soil-dwelling bacterium that you find almost everywhere in the world today. And to our surprise, what we found was that the London strain from 670 years ago actually falls almost exactly basal to the vast majority of strains today, to branch 1, 2, 3, and 4. And that kind of makes sense if you think about it, because it was such a large pandemic, so a lot of diversity got born. So it's a bit like when people in 1,000 years look back at the corona pandemic from 2020, they will also see that at that time, we see a lot of diversity of corona being born, and the same was the case here. We then did follow-up work to see what happened after the Black Death. So this was work that was led by Maria Spirou. She actually looked at many different sites all over Europe. And what she first looked at was a genome that was actually quite interesting. It was from Eastern Europe, very close to Crimea, so very close to the place that we think the disease was actually introduced to Europe. And what she found, if you look at the phylogenetic tree, where we zoom in now with 4 main branches, and here's the London strain from the Black Death, you can actually see that this genome from the Leichenau site, which is in the Volga region there, falls 1 mutation basal to the Black Death. And it falls only 1 mutation away from the most recent common ancestor of all those strains. This is quite interesting, very exciting. We're only 1 mutation away from the most recent common ancestor of the vast majority of strains in the world today. But what is also interesting is that it doesn't have a single mutation that is derived here. It's really basically the common ancestor then that leads to the Black Death. So it really goes from here to there, which also tells us that those strains of the Black Death, as well as this Leichenau strain, have nothing special. They don't have certain genes. They don't even have a single mutation that we only find in them and not in the descendants that you find, for example, then spreading in the 19th century, or the ones you find today in Madagascar, or you find today in the Grand Canyon. They're all going back to those 2 most recent common ancestors, if you want. And that's interesting because if we then talk about the virulence of plague, the virulence hasn't really changed. We would then reconstruct. So basically, if you kind of go to the Grand Canyon today, you find one of those squirrels, or to Madagascar, those strains have the same potential to cause the Black Death than the ones that were around 670 years ago. What she then looked at was a number of genomes that are actually from the time of the Black Death, one from Spain, one from France, one from England, and some from Germany. And surprisingly, what she found was that all of them are identical. So from this 5-year time period of the Black Death, where 50 million people died, all the genomes seem to be identical. So basically, all those people died from a bacterial clone. There wasn't enough time for new mutations to arise. And that kind of makes sense because we have reconstructed the mutation rate now of Yersinia pestis to be about 1 mutation in 10 years. So unlike corona, where we already have dozens of mutations occurring in 1 year, of course, this is a bacterial pathogen which has a much, much lower mutation rate. So it's really only 1 mutation in 10 years. So it was just a 5-year time period. So we wouldn't have even expected much mutations to arise, but it's still interesting to see that all those people died of 1 single bacterial clone. And then we see later on in the late 14th century, new mutations arise. So 3, 4, 5 mutations, and they're all leading like pearls on a string towards this branch 1, which is worldwide found today, as I mentioned, Madagascar and the Grand Canyon, so different parts of Africa and South and North America. And that's also the one that spreads in the 19th century. And that's interesting because we see really the evolutionary steps towards this branch 1 happening in Europe. So what we have reconstructed now seems that the mutations that give rise to this later branch 1 that then gives rise to the pandemic in the 19th century happen all in Europe. So basically the origin of the 3rd pandemic, even though it didn't start in Europe, was probably during the medieval time in Europe. And then Maria looked at the number of genomes from different sites in Central Europe, and what she found was that they all fall on this branch here that is extinct today, so it's not found anymore, but it derives from the Black Death. And this is basically what we have in Europe after the 14th century. So the next 300 years, we have about 7,000 outbreaks of plague, and they all derive from the Black Death. And that's also interesting because people had suggested before that the plague was reintroduced from China or from Central Asia during the centuries, but it doesn't seem to be the case. So it was actually lingering around for those 300 years in Europe. And there were basically new epidemic outbreaks in different parts of Europe, but from a reservoir population that must have been present in Europe at that time. And then the last ones from the 18th century and late 17th century are actually quite interesting because if you look at them in the genome, they have deletions. So there's actually a larger region missing, about 50,000 base pairs missing. And the genes that are deleted in this part of the genome are a part of the flagellum apparatus, So the common ancestor with pseudotuberculosis, the kind of closest relative, still had a flagellum, which is easily detected by the immune system. But through time, it seems that this bacteria have lost the flagellum to evade basically the human immune system. That is still ongoing also during the late part of the pandemic. And some of the genes deleted here are also virulence genes, which is also quite interesting because it seems that it potentially lost a bit of its virulence through time. So then for the Black Death, what we have reconstructed, we have now good molecular evidence that indeed it came from Eastern Europe, was introduced into Europe, then evolved into a strain that moved back all the way into East Asia, then in the 19th century, causing the third pandemic. But what we also wanted to look at now was to see if plague was actually present in Europe even before the historical pandemics. So we do have good evidence now for the Justinianic plague, which I don't really have time to talk about today, but we wanted to see whether plague has already been present in Europe even before the 6th century, because the diversity seems to be older than just 1,500 years. But that's more challenging because if you look into the past, and you don't know what pathogen might be present, you can't really do a targeted approach where you fish out specific pathogens, because people might have died of smallpox, they might have died of some completely different disease. So you really have to use an approach where you can look at everything. But it has been quite challenging in the past, because if you extract DNA from an ancient skeleton, you have all kind of bacteria in there, and it's really hard to assign every DNA fragment to a different type of species from the past. But we nevertheless used what we would call a brute force approach, where we use shotgun sequencing. So all the reads that we extract from an ancient skeleton and try to make sense of them in a kind of more in silico approach using different computer algorithms. Unfortunately, we were working together with Daniel Husson and Alexander Herbig, who developed an algorithm called MALD, an alignment tool, which allows us to assign reads very, very fast to different genomes, different organisms. So basically you can take as a MALD database all the genomes that are available in the world, and you take then your metagenomic sequencing reads and assign them and align them to what species they actually belong to. And this is a super fast algorithm. It's about 200 to 300 times faster than the fastest algorithm that was available before. That allows us to assign about 1 billion DNA fragments in less than 24 hours to different species. And that's super fast, so you can really then take the DNA from many different skeletons in a very short time, assign what type of pathogen might be present in them. And that was then also extended by Felix and Ron. So Felix, a postdoc, and Ron, a PhD student, to then get some extra information and assign, for example, if it's ancient DNA or maybe modern soil-dwelling DNA. And they were then able to look at more than 2,500 skeletons and see what type of pathogen might be present in them. And to our surprise, what we found, and there was also other groups, so from Copenhagen, for example, they found similar things at the time, that Yersinia pestis was already present in the late Stone Age and in the early Bronze Age in human populations. You can see from many different sites. So this is Central Asia, but also from Western Eurasia, from a time from more than 5,000 years ago to about 3,500 years ago. And what we found when we reconstructed in the phylogenetic tree of this very old Yersinia pestis was that it actually falls outside the diversity of modern Yersinia pestis. It's a strain that kind of diverges here about 5,500 years ago from modern Yersinia pestis. And what's interesting and kind of surprising is that those strains are, again, a bit like pearls on a string. So you have the oldest here, second oldest, third oldest, fourth oldest, fifth, sixth, seventh, that are really like pearls. So it seems that one gets extinct, merges to another, then gets extinct, becomes another one. So it's a really intriguing phylogeny. And it also seems to follow a certain pattern. So you have the emergence here that gives rise to number 2 and 3, and 3 to 4, and 4 to 5, and 5 to 6, and then they go back to 7, which is a kind of interesting pattern, which by itself we would probably not make too much out of it, but it also follows the same pattern that people have reconstructed on human movement during that time. So it seems that this pathogen has been moving with people, which is actually quite interesting. What's even more interesting is that there's a large part of the genome missing. In one of the plasmids, there's a large deletion, or it's actually a later on insertion, which is not there yet. And that is interesting because the genes that are in there are responsible for the flea adaptation. So this type of Yersinia pestis from 5,000 years ago could actually not be transmitted by fleas, because it would have actually died inside the flea stomach. So it didn't have the genes yet to survive in the flea stomach. So this could have not caused bubonic plague. it would have caused sepsis because even the close relatives do. But how did it enter the body? So the only good explanation we currently have, it was a pneumonic form. It was actually by droplet infection. So like corona today, it was transmitted between people. At least that's the best hypothesis we currently have. What we also have now is a genome that's about 4,000 years old, again from Eastern Europe, from the Volga region, but that actually falls in the phylogeny basal to the diversity we have today, but on the Yersinia pestis branch. So it does not fall with this kind of earlier form from about 5,000 years ago. And if you look into the genome, it's the green ring here. It actually does not have the deletion that we see in the 5,000-year-old strain. It actually have that insertion. It has the genes that are necessary for bubonic plague, for free adaptation. There's also a few other genes that it has. You can actually see all those 4 virulence factors, the YMT, which is on this plasmid, but also URD, RCSA, and PDE, which are known, very well studied virulence factors, are all present. So this strain from about 4,000 years ago was fully capable of bubonic plague. And we even think that the pattern that we see here seems to indicate that there was a larger epidemic event about 4,000 years ago, which is a very interesting time in human history, because that's the time when we have big turnovers at the end of the Bronze Age, especially in Europe, but also in the Mediterranean and in Eastern Europe, with a lot of empires like the Mycenaeans or the Hittites to actually collapse for unknown reasons. So it could be related to a large epidemic. Hopefully more data will show more information on that in the future. So then in summary, I could show you that Yersinia pestis has been the causative agent for at least 3 historical pandemics. The Black Death has actually very small differences to modern strains. So basically the strains from the medieval time are still around today and would have the same potential. Fortunately, we have antibiotics. So as long as they're not becoming antibiotic resistant, we shouldn't be too worried about them. Yersinia pestis evolved this flea transmission within the last 4,000 years. There is this epidemic outbreaks already occurring in prehistory. So it's really something that has been around for about 5,000 years in human history. There is this influence potentially also on the migration patterns at that time. So when it actually emerges about 5,000 years ago, we also have this, what you might have heard about already, this big genetic turnovers in Europe with this expansion of steppe herders, the so-called Yamnaya expansion, where people seem to move from the east to the west in Europe, but also into the east into Siberia. And maybe the plague was actually contributing here by wiping out large parts of the population, allowing other populations to move into these regions. And the earliest evidence for true bubonic plague, so if you want Yersinia pestis sensu stricto, is actually in the Middle Bronze Age, so about 3,800 years ago. Where do we want to go in the future? I showed you kind of the type of skeletons we have been analyzing currently, which are the purple dots here. You see we have a very strong focus currently in Europe, but there is, of course, much more other interesting populations from the past where we potentially can contribute by identifying ancient pathogens. And this is what we want to do in the future. And by that, I would like to thank Kirsty, Alexander as group leaders that were coordinating most of the work that I presented today, Maria as a postdoc and PhD student, and then Marcel, Aida, and Michal, who have been doing their PhD on this topic, the rest of my department, and of course, our collaboration partners and the people that provided the funding. So thank you for your attention.
Presentation. We have plenty of questions, so I'll start with the first one, if I can catch it. Is the first question, with such high prevalence of plague in Europe, was there any selection happening in humans?
One of the kind of most interesting and pressing questions, especially if you compare it to Luis's presentation earlier on today, we have been looking hard at that. It was actually part of our investigation for the last 10 years, but we still haven't really found strong evidence of selection, which is surprising, maybe interesting that it did not occur, but maybe there is just nothing in our genetic diversity that would provide some sort of resistance or immunity to plague. And it has been also around for 5,000 years. So it could be that the actual selection happened much earlier, say in the early 5,000-year time period or early epidemics and outbreaks. So what we have been focusing on was mostly to compare individuals from before the Black Death and after the Black Death, where we haven't found evidence for strong selection. But maybe we would have to go back in time, so more to 5,000 or 6,000 years ago. Maybe then we would actually find something changing. But one should, however, also say that it was still as lethal probably 600 years ago as it was a few thousand years ago. So it doesn't really seem that there was a really good adaptation mechanism. Otherwise, we wouldn't have had all those outbreaks occurring through the last 5,000 years. So I don't really think that we have good evidence for any good target that was under positive selection in the human genetic diversity.
Okay. Do you think that human-to-human infection was frequent during Black Death?
I don't really think that it was. It has been speculated a lot, and it's really hard to test that based on ancient genomic data. But why I say I don't really think it was so common is because we don't really observe it much in the time period in the 19th and 20th century during the third pandemic. So it was really an exception. There are a few cases, like in Manchuria, for example, there was a big outbreak in a miner colony, but those miners were also living in very dense living quarters where they shared with many, many people the same houses. So those are the good places for pneumonic outbreaks. But otherwise, we don't really have a lot of examples for that. So I think that most of it was bubonic plague. We should keep in mind that the flea population associated to humans and rodents in that time period in the medieval time was probably much, much higher than it is today. So I do think there was a lot of transmission between people and rodents because of really high prevalence also of human and, of course, rodent fleas.
Okay. Any evidence of plague remaining in Europe between the first and the second pandemic?
We have no evidence for that. It's really surprising that the plague disappears in the 8th century. It has been discussed why that could be. Some people have suggested that also the rat population, the black rat population, actually declines. And we have some evidence now from ancient rats that suggests indeed that the rat population declines in the early medieval times. So it could be that this is related, so that there was just smaller rat populations and less chance for this type of transmission, or the reservoir population, whatever it was, that actually harbored the Yersinia pestis during the medieval time just disappeared and only emerged again in the 14th century. We don't actually know what is the natural reservoir. Where did Yersinia pestis hang out during those centuries, during the first and second pandemic? We assume it is rats, but we don't know. I mean, there's now the first data that just has been published last year from Verena Schünemann's group in Zurich. They actually had a rat from the Black Death time that indeed was infected with Yersinia pestis. So we at least now have good molecular evidence that rats indeed carried Yersinia pestis during the Black Death. But were they the natural reservoir, were they dying as much as humans were, we actually do not know that.
Okay, some more specialized questions. Did you find hypermutator phenotypes selected during second pandemic in Europe?
What we actually find during the second pandemic, which I presented, was this, especially in the late second pandemic, we find this type that actually has less mutations, so less virulence genes. So where some of the virulence genes have actually been deleted. And what is quite interesting and really surprising is that during the first pandemic, we found actually in some French individuals from the 7th century, from Lunel, we actually found the same mutation occurring, but it's a very different branch, and it's about 1,000 years earlier. So it seems that there was a convergent evolution. So the region that got deleted with this virulence factors and flagellum-related genes is actually the same and happens twice at the end of the pandemic. So it could also be that just this loss of virulence was actually responsible for it disappearing in the 8th century, but also disappearing in the 18th century. So it could be that it just was too virulent in those rodent population maybe, and that is why it disappeared. I mean, this kind of burnout of diseases has been described also for other pathogens.
Thank you. Another question. Is CCRS delta 32 mutation still associated with plague adaptation?
Yes, so this is a kind of story that had been published some years ago where people suggested that there is this deletion of the CCR5, so a chemokine receptor 5, and which today causes resistance or immunity against HIV infection. And people had suggested that this emerged during the Black Death. But just 2 years later, there was also already a publication that basically deconstructed the arguments that were made in this earlier publication. It was actually a selection event that, if there was selection, it occurred much further back in time. Now we do have direct data from before the Black Death, and we actually see the frequency is as big as it is after the Black Death and as it is today in Europe, where we have a high frequency compared to other parts of the world. This is why people suggested it could have been the plague. So that's definitely not related to the Black Death, and also not to the Justinianic plague, because even in the Iron Age the frequency is quite high. We don't have too much data from the kind of Bronze Age and say early to late Bronze Age. So if the selection occurred there, we don't have the data yet because we don't have enough shotgun genomes. Most of the data is 1240 capture, this capture data, which doesn't have the CCR5 deletion actually as part of that panel. So we actually do not know, but it seems to have been something that occurred much earlier, and there is not even good evidence right now for positive selection on this deletion, but it's certainly not because of the Black Death.
Okay, thank you. A last question, which is a very odd one, I would say. Is it possible to see in one's genetics if one's ancestors have survived a plague pandemic?
Yes, that would only be possible if indeed we would have found some sort of selection on a certain gene, like Louise presented this morning, where we have, for example, sickle cell anemia. So people that carry that gene are basically descendants of people that probably survived malaria because they carry this gene. So this would be a signature of basically survival. But of course, over 600 years, each of us has more than 1 million ancestors. So we are really kind of a big, of course, potpourri of people that lived afterwards. So you can't really try to trace it back, to a certain group of people. But if there are those genes under selection that give you immunity, those would be then those kind of signals. But for plague, we haven't found it yet. Unlike for other diseases like malaria, where we have very good evidence that there are those immunity genes that provide larger immunity. But there is also often, as Louis said, balancing selection on those genes, where sometimes in certain circumstances they're good, in certain circumstances they are bad. Like for the CCR5 deletion, it's good for HIV, but it's actually bad for West Nile virus. So if you have that deletion, you're much more likely to die of West Nile virus, but you are resistant against HIV. So there is a reason that this gene exists. So it's better not delete it, because it might actually kind of make you prone to get another disease in a much more severe case, like has been shown also for mutation on the chromosome 3 that has been inherited from Neanderthals to us, but gives people a much higher likelihood to die of COVID-19, which in other circumstances might actually be beneficial. That has just been published. So it's often balancing selection on those immunity genes.
Okay, thank you for giving us some light on all those different aspects of plague. Thank you for your presentation, and we'll proceed to the next speaker, who is Dr. Pascal Gerbeau. Dr. Pascal Gerbeau has been working at University College in London and recently moved to the University of Geneva in the anthropology unit. She's an evolutionary geneticist, mainly studying how humans have adapted to various pathogens. And she focuses now in our talk on some examples, and she will address this burning question, trying to know if our species is still evolving. So, Dr. Pascal Gerbeau.
Thank you. Thank you for the introduction. So let me just... Sorry. Yes, I just need to share my screen. Which you should see now. Is this okay?
Yes, yes, we have it.
Great. So thank you for the introduction. So as we saw yesterday, this is a very simplified model of human evolutionary history. So our species originated about 300,000 years ago in Africa, and over the past 100,000 years, humans spread across the globe. The first anatomically modern humans in Europe, as Mr. Evelyn said yesterday, has been pushed back to 46,000 years ago. And we have early modern human genome sequences for humans between 45,000 and 24,000 years ago in Asia. And the peopling of the America across the Beringia here dates back to between 15 to 23,000 years ago. So after humans spread out of Africa, they encountered archaic populations and interbred with them. And this integration represents between 1% to 6% of modern genomes, depending on our population ancestry. Also, this is a map. This is just one of the representations of climates, and it's Köppen's representation. What I wanted to show here is that you have a wide range of climates, and Luis mentioned tropical rainforest as one of them. But as humans spread across the globe, they had to survive environments they had not known before, including extreme cold in much of the Americas and Eurasia during the last ice age, altered exposure to sunlight, and as Johannes showed, pathogens not previously encountered. And all of these induced a wide range of selective pressures. And in the last 10,000 years, most human populations have transitioned from a hunting-gathering lifestyle to producing food, which resulted in rapid population growth, increased population densities, increase in infectious diseases, as we saw as well, and dramatic dietary changes, as also was mentioned earlier. So in the context of this conference, we're looking at the last 50,000 years ago, where we have spread across new environments and adopted a new lifestyle, which have shaped us through adaptation. And I'm going to specifically talk about 3 of these adaptations that I've been involved in the research. So the first example is an adaptation to an Arctic environment, where humans have adapted to cold climates and a lipid-rich diet. The target population here is the Inuit, living in today's Alaska, Canada, and Greenland. And they have a marine-rich diet in omega-3 polyunsaturated fatty acids, also called PUFAs. And the Greenlandic Inuit genomic diversity was compared to Europeans and Chinese using a specific population genetic statistics called the population branch statistics, which focuses on tiny differences between populations. So you have to remember that our species, the different populations, were pretty much similar when we look at our genetic diversity. But when you focus on specific differences between these 3 populations, so the Greenlandic Inuit, Europeans, and Chinese, the most differentiated region was observed to be for fatty acid desaturase enzymes, also called FADs here, which are involved in metabolic fatty acid composition. And this makes sense since they're important enzymes involved in the balance between short and long chain fatty acids. And because of Greenland Inuit diet, they consume very high amounts of long chain PUFA fatty acids, and therefore they need a high activity of these FAT2 gene variants to metabolize these fatty acids. So we run models of the Greenlandic Inuit population demographic history to infer a time and a selection coefficient and a time when selection may have started. And we found that selection may have started anytime between 3,000 and 23,000 years ago, and with a selection coefficient between 1 to 20%. What was interesting was that 2 of the variants, as in the FADS region, significantly associated with short stature in Inuits, and looking at the other populations, we found that these variants were also associated with height in Europeans. And this highlights one of the strengths of these population-specific studies, that if we hadn't studied the Inuit population, we wouldn't have found these variants associated with height in Europeans. So they allow us to understand better genetic diversity. The second adaptation I'm going to mention is the adaptation to dairy consumption. And with the advent of cattle domestication in the Middle East and North Africa about 10,000 years ago, there was a strong selection for the ability to drink milk as adults, which is called lactase persistence. So on the right here, you've got a representation of the lactase gene, abbreviated LCT here. And the variants at the bottom of this representation show the variants that are observed and used to describe the genetic diversity between populations in this region. And some of these variants show some of the strongest signals of selection in the human genome. So lactase, the gene LCT encodes for the enzyme lactase, which breaks down lactose in milk, and the lactose is a big sugar which cannot be absorbed into the bloodstream into 2 smaller sugar, glucose and galactose, which can both be absorbed into the bloodstream. Now, what you have, the diagram on the right is hand-drawn just to illustrate what I'm saying. So mammals, including humans, have lactase, and lactase activity is high at birth and decreases after weaning is over. And that's also the case in most human populations. And this trait is called lactase non-persistence. However, some human populations have evolved the ability to express lactase throughout their lives, as is shown by the green line here. And this continuous expression of lactase throughout adulthood is called lactase persistence. And it allows to digest milk and the lactose in milk without symptoms of lactose intolerance. So here, they are the variants that are associated with lactase persistence. And so blue here shows absence, and yellow shows high frequency. And you can see that in Africa, you've got all these 5 variants represented, but in Europe, there's only 1 of them. And in fact, this variant is the one we know most about. So we also have ancient DNA data for this allele, for this variant associated with lactase persistence in Europe. We don't have that much ancient DNA data for Africa because of ancient DNA conservation issues. But what these maps show is that you can see this allele represented in blue here, increasing in frequency through time. So from ancient DNA record, we know that this variant associated with lactase persistence was relatively rare before 5,000 years ago. And we know that dairying, there is archaeological evidence for dairying in the Near East from 8,000 to 9,000 years ago. So I've represented here very schematically population demography, where you've got the past at the top and present at the bottom. And if this was human, if the blue was representing human population expansion, you have agriculture and dairying here, which are very recent innovations in relation to our evolutionary history. And the allele associated with lactase persistence would have occurred very recently, and in Europe would have spread and be present at very high frequency in some Northwestern Europeans. And this genetic pattern can only be explained if we infer natural selection has been at play. And in fact, depending on the methods used, we infer that the selection coefficient on this variant is one of the strongest on the human genome, at least in Europeans, over the past 20,000 years. And similar selection strength for major African variants have been obtained as well. Why this variant may have been selected for, why lactase persistence may have been under selection, we don't quite know. The reasons are still debated. And so if you have any suggestions, they'll be welcome. The last example I would like to talk about is a variant that is observed at very high frequency in East Asians, and it's located in the ectodysplasin A receptor locus, also EDAR. It is significantly associated with hair thickness and short types incisive. When we run models of population evolutionary history to infer the selection strength and the time when selection may have happened, we found out that selection may have started in a time between 13,000 to 40,000 years ago, with a selection coefficient between 3 and 19%. But the main reason why this study is so exciting is because a humanized mouse model was produced. So you can see here on the left a homozygous with the not carrying the allele, a heterozygous individual in the middle, and a homozygous carrying the variant on the right. And the hair thickness in these mice were reproduced, which was really exciting. The shovel type instances were not, but that's probably because rodents may use their teeth differently as humans. So an interesting aspect with mouse model is that you can use them, you can look at different phenotypes that you may not be able to see in humans. And so these mice had increased sweat gland densities. And when we looked at a cohort of humans carrying this variant, this was also recovered, which was really interesting. And then these mice also had a decreased mammary fat pad and increased mammary gland densities. So this shows that with humanized mice models, you can explore various effects of a gene and its pleiotropic effects. So I've only shown here 3 examples of recent human adaptations, but there are quite a few as highlighted on this map. And it's quite challenging to establish a complete understanding of human adaptations because we first have to identify the genomic region under selection. As Johannes also mentioned earlier, the phenotypes that selection is acting upon and the external conditions that have driven this selection. In addition, and as Luis mentioned as well, many genes affect more than one trait. They're pleiotropic, like EDAR, and many traits are encoded by more than one gene. They're polygenic, such as height. So identifying these specific adaptations remain a challenge. In the context of this this conference, we've been focusing on the last 50,000 years, and the answer to the question, are we still evolving, is yes, that's clearly the case. And in fact, we can't stop evolving. I have shown some examples of adaptation that have occurred within the last 50,000 years. But there are now population databases such as Decode and the UK Biobank that are databases with a collection of phenotypic and genomic information for thousands of individuals, and they allow the identification of selected traits within a few generations. So we sometimes think that evolution is towards something better that we have, and we have these idealized versions of ourselves, but they're often very cultural specific and often fantasies. Natural selection doesn't really care for something we would find pretty. It works differently. So I'd like to take a few minutes to thank you for your attention, collaborators involved in this research, and the organizers of the Académie des Sciences for organizing this great conference. Thank you.
I may have one. Someone has a question in the audience here? No. Yes, Luis.
I would like to know if you can...
Can you hear? Can you hear the question?
No, I can't. I'm sorry.
So I'll have to repeat probably.
Can you come to the microphone?
Microphone.
Hey, I would like to know if you can clarify the adaptive nature of the EDAR mutation in East Asians, because I know that it's related to teeth morphology and hair thickness, but I cannot see how this can be adaptive. So there must be an underlying phenotype that the genes of this phenotype are pleiotropic, and incidentally you get thicker hair and different teeth shape. Thank you.
Yes, thank you. This is as the slide showed, the mouse model allowed to identify other phenotypes that this gene was affecting, such as the number of sweat glands, their density rather, and the mammary fat pads, the mammary glands as well. So these may be the adaptive traits. However, we don't quite know yet. So it's also still debated.
Question.
Yes, I have a question about lactase persistence. How do you explain that? I mean, if this is under a strong positive selection, how do you explain the discrepancy between the development of agriculture in Europe and the development of this selection? But it looks like it's mostly, let's say, at the eve of the Bronze Age, or even at the beginning of the Bronze Age, that this mutation becomes prevalent in Europe. What's going on during the Neolithic? They don't use milk, or what?
Archaeological evidence does show that Neolithic population use milk because there are sieves that are used to ferment cheese that have been found, and also lipid residues show dairy fat residues in various potsherds, which is why we know that Neolithic populations were using milk quite early on. But it's true that we don't see this allele at appreciable frequency before 5,000 years ago in ancient DNA. So it's possible that it's been brought in from other populations, because we know that there has been quite an important population turnover in Europe at the Bronze Age. Again, whether it's a demographic expansion and integration, or whether there has been an important selective pressure that has favored the ability to drink a lot of milk without having symptoms of lactose intolerance, we don't quite know yet.
Another question concerning nutrition. What about the microbiota impact evaluation?
Impacting? Sorry?
About the microbiota is there any clue about the effect of microbiota over time regarding nutrition? So
I'm not sure I understand the question. If the question is about the importance of the gut microbiome in relation to the tolerance of lactose?
Concerning nutrition more generally?
I'm sorry, I don't understand.
Okay. Other question, did all the lactase persistence alleles rise up in frequency around the same time? Or did selection on these occur at different times?
So the selection intensities that have been inferred are about the same, and because the signal we see is very similar, then it seems that they have arisen at about the same similar times. But we don't have as much ancient DNA information for the parts of the world than Europe. So this will probably tell us more about when these other alleles have been selected and appeared.
Another question about the lactase persistence is could lactase persistence be linked to adaptation to famine, for instance? Famine.
Yeah, I don't see why not. It's possible that if there wasn't any food resource available so we know that early Neolithic populations were trying on new things, and as they were migrating into new environments as well, because the crops they had may have been adapted to a warmer climate in the Near East, and they were transporting these crops in higher latitudes, and so there might have been failure of their crops, and they had to consume their animals' milk instead. So it's possible that it brought a good complement of nutrients then in these situations. You can easily see why lactase persistence in a situation when you can't have access to a pathogen-free water source can be important. So one of the symptoms of lactose intolerance is diarrhea, which is not as problematic in our cultural context, where we can drink lots of water. But if you don't have access to a free pathogen, a source of water free of pathogens, then that may be very bad, and diarrhea is actually lethal in some parts of the world as well. So yeah, definitely that's a possibility.
Okay. Maybe a last question about the examples of frequent deletions of CYP450 genes in some population. Are these environmental adaptations, or?
I'm afraid I don't know. Maybe some other members of the panel who talked today may be able to say more about these specifically.
Non-answered question. That's okay. Thank you very much. We'll now proceed to our last speaker this morning, who is Evelyn Heyer, who is here with us. So she works at the Muséum National d'Histoire Naturelle, where she's professor in genetical anthropology. She, among others, is mainly a field worker, and she carried out studies in Africa and Asia. She's also strongly committed to the public outreach of science, and recently published a book that maybe some of you know, which is L'Odyssée des gènes. I didn't read the book, but some of us did, and Bernard Dujon told me that it reads extremely easily. And so I'm sure that it will be of interest for many of the audience of this meeting. Now, coming back to one of our main research topics, she will explain to us how culture impacts genetic diversity. So thank you, Evelyn.
So thank you very much for this introduction. So the focus of my topic is how culture can impact genetic diversity. So first, I want to recall you that our species has a very high level of cultural diversity, and that I use for culture the definition of the UNESCO that include not only material culture, but also lifestyle, way of living together, value system, tradition, and beliefs. So a big, huge definition of culture. So the question is how these differences can impact our genetic diversity, and with what mechanism. So the first mechanism that has been shown by the previous speaker, Pascal Gerbeau, is that it could happen through culture and selection through what is called the niche hypothesis, the fact that we change our environment and in response we have genetic adaptation to this new environment. One of the major examples is of course adaptation to diet, with the most classical example being the lactase enzyme that gives the possibility to adults to digest milk. And it has been shown quite more than 50 years ago that it's frequent in pastoralist population. So the idea is that indeed by a change of way of life, I mean using dairy product, we induce a selection in our gene for this lactase persistent mutation. And indeed, lactase persistency show a high diversity of frequency across the world that correlate more or less with the lifestyle, but with very strong exception. For example, Central Asian pastoralists who have a diet rich in dairy product show a very low frequency of lactase persistency gene. It's what is presented on the right on the graphic, where you see the frequency in farmer and in pastoralist, and indeed in Central Asia, the frequency of the lactase persistency gene is very low. That raises a question of another type of adaptation, either through the microbiome or through other cultural practice like fermenting of dairy products. Other examples that have been presented is the FATS1 genes in the Inuit for lipid biosynthesis, or the FATS2 gene in South Asian in vegetarian population. Indeed, one of the major change in our way of life is the Leolithic Transition that occurred 10,000 years ago, where we changed from hunter-gatherer nomadic way of life to different way of life, like pastoralist or farmer, and in some cases it's lead to strong imprint in our genetic by adaptation to new diet. Regarding lactase persistence gene, as you have seen previously, the date when selection started is still not clearly known, because indeed when a gene is under selection, it takes a lot of generation before it increases in frequency. So it could have been selected at the Neolithic transition, and we can only detect it at the Bronze Age when it reached frequency high enough. So this is still a matter of study. But beyond this cultural selection, we have several other cultural practices that can have an impact on our genetic diversity. The first is who we choose to reproduce with. We know in human population that the major component is geographic distance. We tend to marry with someone who is close geographically, and this leads to a gradient of genetic diversity that follow geographic distance at a global scale. But other phenomena where culture can have an impact have been detected. First, in the case where the choice is according to a cultural trait, and this trait is transmitted from one generation to the next, it can lead in only tens of generations to genetic differences according to this trait. To be more concrete, I show you an example of this case of language in Central Asia. In Central Asia, you have two groups of population that have different way of life and languages. The farmer, who are Indo-Iranian speakers, and the Turkic speaker, who are nomadic herders. And by doing field work where we sample DNA, but also linguistic data, we were able to compute linguistic distances among population, and we show that indeed there is a strong genetic linguistic correlation. People who speak a similar language tend to be similar genetically, and we are in a nice case study because there is a very low genetic geographic correlation, which can be a compound that explains the genetic linguistic correlation, but it is not the case. We clearly have an impact of language on genetic in this area of the world. In fact, language can reduce or facilitate migration exchange. You tend to marry with someone speaking a similar language. Not excluded that language can be a correlation with other cultural aspects that could be important for mate choice. Beyond language, that has been the most studied trait, there is religion, caste system, for example, in India, where we show that cultural practice can create genetic differences among population. Indeed, if this cultural endogamy tendency transmitted in only tens of generations of local endogamy can generate slight genetic differences that can be detected with genome-wide diversity. Recall it's a tendency. You never have close population that practice 100% endogamy. A second aspect where culture can have a strong impact is indeed in admixture. It has been shown previously that several times during history we have divergent populations that came into contact and tend to exchange genes, like the Neolithic and local hunter-gatherer people, the Bronze Age steppe with the local Neolithic population, the more recent example of admixture following the slavery trade, where you have sex bias admixture. Indeed, admixture in most of the case in human population is something under control of social rules. As a very good example, I will show you the sex bias admixture between what is called pygmies and their neighbor non-pygmies population. Luis Quintana already presented the pygmies hunter-gatherer. Indeed, here is a map of the different pygmies population in Central Africa, where we have designed field samples for more than 10 years, and that had led to a lot of wonderful study in our team and with Luis Quintana team. But I focus now more of the social constraint of admixture. If you look at the left of this graphic, it shows you how pygmies admix with non-pygmies population. Indeed, the only marriage possible is between a woman pygmies and the non-pygmy neighbor population. Due to patrilocality, the pygmy wife will move to the village of the non-pygmy and she will get offspring, but because of ostracization or other social factor, she will come back to the pygmy village, so that if you look at the right, this has clearly asymmetrical consequences. If you look at the female lineage, in pygmies population in red, they have what we will call the pygmies haplotype. Conversely, regarding the male lineage, in several pygmies population, you have a complete replacement of the Y pygmy lineage by the non-pygmy Y chromosome. And this is an example where you have clearly a Y chromosome replacement without any violence, only because of social constraint in the marriage that led to sex-specific migration. The third component in who we choose to marry with is what we call kinship system. We all live in society where we have a kinship system that has three components: the residence rule, the alliance rule, the descent rule. So this leads to a huge variety of different ways to make family in our species. Regarding the first component, residence rule, that is the question where you will settle after two individuals marry from different villages. In patrilocal population, the couple will move to the village of the husband. In matrilocal population, it's the man who will move to the village of the woman. It's what is represented down, patrilocal versus matrilocal. So in patrilocal population, the woman move. In matrilocal population, it's the men who move. So for example, in Central Asia, if we look at these couples, we see in red the high frequency of patrilocal practice, with more recently what is called neolocality, where you don't go to either village, but you want to go to another place rather than in the family of the father or of the mother. 64% of the society are patrilocal. And this migration has an impact on the genetic diversity. So, for example, in the case of patrilocality in Central Asia, the genetic differences amongst population are much smaller for the mitochondrial DNA than for the Y chromosome. Indeed, women have a high level of local migration, but men much smaller, so that they keep a very high differences for the Y chromosome between population and ethnic group. The second component of what is called the kinship system is the alliance rule, who you choose to marry in relation to kinship. And it has been widely shown that in some population you have close cousin marriage, whereas in other population you don't have it. And this change the genetic consanguinity, as it has been shown in this worldwide study. The third component of kinship system is what is called descent rule. It determines the family you belong to and your place in the social group. You have patrilineal population where you belong to the family of the father, and matrilineal population where you belong to the family of the mother. You have also some society, like in Europe, where you are indifferentiated, what is called also cognatic. You belong to the two families. You know your four grandparents, also from the father and the mother side. Descent rules vary across the world. So you see in yellow the matrilineal, in blue cognatic, and in white patrilineal. There is a huge diversity across the world. And for example, in some population in Central Asia, you have patrilineality. So individuals belong to a same lineage because they have a recent common ancestor through the father. Several lineage form clan because they claim to have a recent common ancestor through the father line. You belong to the lineage, the clan of the tribe of your father, and it's transmitted from one generation to the next. So it's an example of genealogy in the Kyrgyz village stone hall. It's something very present in this population. When you ask someone who he is, he will tell you all about his lineage, clan, and tribe. And this has an impact, of course, on the Y chromosome diversity. It increases differences among population, and it reduces diversity inside population. But indeed, the three components, alliance, residence, and line of descent, form what we call a kinship system. And one of the researchers in the lab study in detail the high diversity of kinship system in Southeast Asia with matrilineal, matrilocal, patrilineal, patrilocal. And what she saw that is very interesting is that beyond an impact on the mitochondrial DNA and the Y chromosome, it has also a strong impact on autosomal genetic diversity, for example, with a higher inbreeding level in matrilineal population. So the third component, so the first one was culture and selection, the second is who you choose to marry with, but the third one is what do we transmit. And we developed some research of what is called the cultural transmission of reproductive success. Indeed, when you look in details of family, there is, for some families, a tendency to have a high number of children, and their children will have a high number of children. Conversely, you have family where you have a small number of children who will have a small number of children. We talk about effective children. That means the children who transmit their gene, who will reproduce. If you have 10 children but they do not reproduce, you have a zero number of effective children. And this cultural transmission of reproductive success run in our species for several reasons. First, there is a tradition that runs into families that has been well studied by demographers. You have a tendency to have big family, and you will want to have also big family on your turn. Second, there is what is called cooperative breeding. You know that in our species we have what is called allocare, so that it's not only the father of the mother who raise a child, but the extended family has also strong impact on the raising of the child, so on his survival probability and probability to reproduce. So the larger is your family, the higher number you have of sister and brother, the higher will be your number of descendants, your reproductive success. And third, in humans, there is transmission of cultural traits that are linked to reproductive success. That has been first shown in Amerindian population, where high-ranking male are more polygynous, so they have more females, so they have a higher reproductive success, and they transmit their high-ranking social status to their son, who on their turn have also more female and more reproductive success. You have another example in Maori population where its female high status increase the survival of their children, and they transmit to their daughter their high status. In eastern Quebec, it's in relation to immigration. You have huge family size, but in some family you have only a small number of children who remain in the population, and in other family, most of the children tend to settle in the population, and at the next generation, they have also a lot of children who settle in the population. So these have several genetic consequences. It reduces the genetic diversity inside population, and also it explains in some populations the frequency of some genetic disease that could have been eliminated by natural selection, but they are each hike by a form of cultural advantage. So it's the case, for example, for the disease in East French Canadian, where you have a high frequency of some inherited disorders, but more closely, it's also the case here in what is called Jura, in the Valserine Valley, where because of this cultural transmission of reproductive success in relation with land property, when you belong to the village, you have more children that settle because you have lungs because you have properties, and these children also will transmit this advantage to their children and so on, so that you counter-selected a gene that should have been eliminated by natural selection and that remain in high frequency in this area. So all these cultural traits are not independent. In Central Asia, you have patrilocality, patrilineality, linguistic endogamy, male transmission of reproductive success. In our species, there is a high diversity of norms that are more or less followed and also transmitted. So there is a high level of cultural diversity. But for cultural diversity to have an impact on genetic diversity, you need these norms or rules to be transmitted, which is the case of several cultural traits. So cultural diversity has an impact on genetic diversity, but the mechanisms are highly diverse. And this is very important because it's a paradigm shift in the fight against racism. Rather than genetic differences determining our cultural identity, we see that it is cultural identities that often influence our genes. So thank you very much, and I thank all the team and the people who are collaborating with.
So thank you for this a very nice and very clear presentation with plenty of questions. Could you comment on the scientific value of the results for DNA tests that are performed by companies like 23andMe and others? What is your view of practicing those tests, the pros and cons?
Okay, so I will not go about the fact that sometimes it's difficult to know how accurate they are. Okay, first, I think all these companies work very well, but they don't give us the data to be able really to give a scientific assessment of their results. I think that it's the right to anyone, if he wants to know his past ancestor using genetic data, but he has to be very careful. First, when he tests his DNA, it's not only his DNA that is incorporated his databases, but indirectly all the DNA of his close family. So it's something that really has to be taken into account. And second, there is a risk that this genetic defined identity become more important than other identity. And for me, I think that your cultural identity should always prevail from your genetic identity. It's not because it's scientific that it has to have more importance in what people can define himself.
Thank you. Second question. How do populations who practice cultural endogamy deal with health issues? Do they know that this is not the best way to avoid disease?
I mean, cultural endogamy, I would say it's not, as I say, it's something very, you still have exogamy, a little exogamy. So you still have in migration, it does not exist a population that is completely close to other population, first. And second, in all population, there is avoidance of marriage with close kin, so it's not really a problem.
Okay. Now we have some questions from other speakers, from Louis, for instance. What about cultural endogamy in royal families, like the hemophilia transmitted in Queen Victoria, descendants, etc.
In this case, they did not avoid close kin, so we see indeed that it has really a problem with the occurrence of several recessive disorders because of close inbreeding.
Okay. Well, this is even a discussion between speakers. I'm not sure I understand everything. Jean-Louis Mandel claims that hemophilia being X-linked is not dependent on endogamy, but on associative mating between prince and princesses.
Yeah, I agree. I mean, you have cultural endogamy. For me, it's when you tend to marry in the same group because of the cultural trait. So I will say regarding all those families that it's more close kinship in order to keep power that was at action rather than what I will call cultural endogamy, because yeah, I completely agree.
But you can...
Just a question about this. How
Microphone. Microphone.
I will repeat the question.
How do populations who practice cultural endogamy justify this culturally? Which is the way, I mean, why I have to marry a first cousin? What's the good on that?
Okay, the question of Ruiz is what populations say about cultural endogamy, how they justify it. I mean, there is a tendency in human to differentiate culturally from one group to another. It's a clear process that leads to cultural diversity in our species. And in some cases, this idea to differentiate from other is translated also in cultural endogamy. That means the fact that we prefer to marry someone who, for example, speaks the same language. And it's something that's very strong. For example, I presented part of the result in the link between language and genetic diversity in Central Asia. I presented it in Kazakhstan, and when I presented it to the people, I told them, so it seems that you prefer to marry someone who lives 1,000 kilometers away from where you live, but speaks the same language, rather than marry someone from the neighbor village, but we speak a different language. And they all agree that for them it was something clearly important, and they would rather choose someone 1,000 kilometers apart than marry someone from the neighbor village, but who speak a different language. But as I recall, I think the choice is not only language, is that language is a way to measure this cultural diversity, but it could be for other cultural traits that are more important than language. For example, way of life, way of behavior, and things like that.
One part of the answer to the question is also given by one of our participants. It's Jeremy Chouin, and he was saying that it was a way to keep goods within a family.
That's the case, for example, for the very nice example of Raphael Scheck's about the matrilineal and matrilocal population. Indeed, it's the husband who should leave the village to marry outside. If he marry a wife in another village, he will have to move. So he will choose rather to marry inside the village in order to keep the control of the goods. So indeed, he's right. In some cases, it could be the way to keep goods. But what I want to stress is that in humans, there is a huge diversity of cultural practice. And all the study we have done in the lab, it really emphasizes this huge diversity. So you don't have one rule. So for example, in Central Asia, the patrilocal, patrilineal are extremely exogamous, and where the other population are extremely endogamous. And it's certainly not for the same reason that you have these differences in Southeast Asia.
Question from Jean-Pierre Changeux. The balance between cultural belief and rationality.
I really like Jean-Pierre Changeux, but I think it's something I really would like to discuss with him. It's a very huge topic, and it's something very interesting. But I think it's beyond this conference, or it will take much more time to discuss this.
Okay, there are a few other questions, but I'm not sure we are going to take them. Maybe exogamy is the way to keep maximum of distance with your mother-in-law.
Yeah, we don't know why some society are patrilocal or matrilocal or neolocal. We don't know. I mean, there is a lot of people have tried to find rules about this cultural behavior, and there is no convincing evidence that say why you will move or not, and why the female will move or the man. It's under debate, I think.
Okay, if we have no other questions, I would like to thank the speakers for this very interesting session we had this morning. I think we can applaud.